organizers, and most importantly to the attendees who have taken the time out of your day to join this conversation. So in previous discussions, we've provided an overview of some of the molecular technologies, next-generation sequencing in particular, and the ways in which that's changing the way drugs are developed. Today we're going to take a more focused look on how these tools are being utilized specifically to provide greater insights into tumor biology. I will first introduce the EA laboratory and then provide an overview of the next-generation sequencing tools that have been developed there, and then hand it over to Dr. Bacus to review how the resulting data from these tools are being used to elucidate the mechanisms of cancer biology, discriminate between the driver and passenger mutations, and then delve specifically into the case study of inflammatory breast cancer. So with that, EA Quintiles is a genomic services provider that was established in 2001. It was first spun out of the Duke Microarray Core facility, headquartered in Durham, North Carolina, as an Affymetrix microarray provider. And through that time, we've really developed a KOL leadership position in the area of innovation, working with the FDA on standards, and developing operational excellence and expertise to deliver cutting-edge data to its customers in the area of genomics. As technology evolved, we became a provider of next-generation sequencing platforms in 2011. And with that, we've maintained our thought leadership position in both the bioinformatics and, again, in the area of being able to compare data between different organizations, different studies, etc. So we were the first CLIA-certified lab to offer GLP-compliant microarray services, and again, we've taken that into the areas of next-generation sequencing. Overall, our goal is to be able to provide end-to-end solutions from the nucleic acid or even the samples that you provide to us. We can take it through all the way to bioinformatics and being able to provide you the answers to your biological questions. And this is done in a multitude of different technology and applications. So really, I should mention, sorry, that EA was acquired by Quintiles in 2011, really as a strategic move to be able to support the integration of genomics into the clinical development process. And that's what the next slide highlights, and where the different technologies and applications are currently supporting the clinical development process. So technologies such as RNA sequencing or exome sequencing, as well as microarrays, provide very early hypothesis generation avenues for investigators to start to understand which pathways may be tied to their particular therapeutic. Again, bioinformatics is the cornerstone of helping customers be able to get to those types of hypotheses. We can then take it a step further by providing many of the targeted genomic and expression assessment tools through both EA and some of the laboratories within Quintiles labs, which are able to provide other applications such as immunohistochemistry, FISH, etc. But what we've seen as of late is that there's a trend towards next-generation sequencing and being able to look simultaneously at many different targets within a particular patient sample and starting to understand how nucleic acid changes can impact the resulting response to a particular therapeutic or safety signals, etc. And with that, we've developed a few tools, which we'll speak to in the next slides, which are able to assess handfuls of genes that are thought to be relevant to a particular customer's interest. So with that, I'd like to introduce the clinical sequencing solutions that Dr. Bacus will speak to how we've utilized those tools in her portion of the presentation. So to start with, EA Quintiles has extensive experience in many different platforms, and we remain agnostic to which platforms we would recommend to a particular customer. But it's through this experience that we're able to gain the insights about different platforms and make really solid recommendations to enable the best technology for that particular customer. So here we're listing some of the different platforms we've tried for sequence enrichment or selection, and that is to be able to tease out the regions of the genome that are applied to next-generation sequencers for readout and assessment. Beyond that, we've been able to look at many of the different sequencing platforms, and we've employed many of these in our production line. In fact, all of these listed here are currently in our production line. So we've utilized Illumina Technologies, Life Technologies, and Pacific Biosciences. And in previous discussions, we've highlighted some of the key benefits or disadvantages of the different technologies, but we're always happy to speak with you about some of those different advantages and make recommendations for you. But to summarize, we've used these different combinations of technologies to enable over 20 different custom designs, primarily in oncology, to look at specific gene lists. And we've had the opportunity to process over 5,000 samples for our clients using these different designs. In total, we've been able to design panels for many different target region sizes. So in some cases, we may have clients that look at one gene. Some clients may want to look at hundreds of genes. And we, again, utilize the right technologies to apply that to those scientific questions. And in some, we've been able to look at 3 kb up to 3.2 megabase target regions.